if you were to come again to a workshop like that, what you would like to see there. All right, so a little bit practical information. So I'll be talking for another probably 10 minutes, and then Shawn will take over. So we ask you all to bring laptops with you, because that's what you will be using to go through the tutorials and learn how to do stuff. So at that point, we had basically made a difficult choice, right? We could have asked you to install all of the software that you need to your laptops, or we could have used Amazon Web Services. So we think we'll do mostly second, although you can install things on your own if you like. I think Shawn has a very good instructions how to do it on your laptops. But if you want to have Cadnano or a new Cadnano, so to say, on your laptop, you can do it. But we also made an image at Amazon Cloud where all our software is available. So all of the software, all of the tutorials is there. It has a little bit of a pain to get to it. We will have a tutorial on that. It's 20 minutes to set everything up, but then once everything is set up, you can actually run stuff there, because those nodes that you will get access to, they have a proper graphics card with good accelerator. So unless you have a gaming laptop, you won't have a nice graphics card. But through AWS, you will have access to it. So it's kind of a little bit of trade-off. So we'll see how it works. Okay, so just a brief overview of the tutorials that we will offer, and you will have a little bit more description of it later on as we go on. So you can say that conceptually we are teaching two things. One is we're teaching design and some simulations with Cadnano and Cadnano toolkit. So you'll have a lecture on Cadnano today, and tomorrow Troll will talk about the Cadnano toolkit. At the same time, we also want you to learn how to do molecular simulations. I'll give a lecture on all-atom molecular dynamics simulations of self-assembled structures. And there are two tutorials that go with it. I'll introduce them in my lecture. But then tomorrow, we also have another lecture that Chris Matthew will give on how to use multi-resolution tools to simulate complex origami structures. And so that's kind of the simulation track. And somewhere in the middle is we have a tutorial on how to make images and movies using VMD Pro specific to DNA structures, right? So at the end of the day, everyone wants to publish a paper and give a talk and have some cool animation. So we will teach you how to do that. Okay. All right. So here's the program. The program is has a very simple layout. We have two lectures in the morning, coffee break and the third floor, and a lunch break also in the third floor. So that will happen, and afterwards we'll come back here after lunch and we do hands-on stuff. So the first thing that we'll do, we'll set up AWS, and we will do it like step by step all together. There will be TAs to help you to go through it. And then basically you can choose which tutorial you want to do. Because it really depends what you already know, because some of you are already proficient users of Cadnano, or some of you know how to do microdynamic simulation. So you can mix and choose and mix what you want to do. The suggested order is that you do Cadnano tutorials today and all-atom tutorials today, and others you do tomorrow. But again, if you already know something, you can move on to the other one. So we will have a dinner today at 7. Can I raise your hand if you're planning to attend the dinner?
The food is on us.
Yeah, the food is on us.
The alcohol is on you. But the food is on us. I think probably.
Yeah. So okay. So it looks like everyone. So very good, because we have to finalize one. So it will be after 7. So you're welcome to stay here as long as you wish and go directly to the restaurant, or you might want to go back to the hotel. And it's really up to you. And later on, I actually show where the dinner is, with the map and everything else. All right, so yeah, so Shawn will talk about Cadnano right after me. So tomorrow we have basically the same setup. So Tyrell will talk about Cadnano Toolkit. It's a new stuff in Cadnano. And Chris will introduce this multi-resolution method to simulate origami. I think both will be excellent. And complementing, you will have tutorials. So you'll have tutorials on multi-resolution DNA simulation package. Now, some of you might already be in the field and probably working on some particular problem. And it actually happens quite often at this workshop that people come with their own systems and say, okay, now I know how to do your tutorial. Now, can I do it for my system? Sure. So please, once you go through the tutorial and you understand how it works, you're welcome to try to do it on your particular system. And we will be here to help you to go through it. So in effect, you can have your project done basically by Wednesday. Everything goes well. And on Wednesday, we have even less lectures in the morning. It will be just introduction to how to make movies, really like what you can do with D&D, and also how you can use other software, how you can integrate what you do with other software. So predominantly we focus on integration with Cadnano, but there are other tools around, and we are supporting all of them at this point. So that is it, I think, for me. So let me do some introductions. So Shawn Douglas is here. Tyrell, can you raise your hand? It's over there. And so that's UCSF team, only 2 people. And Illinois Williams here, we have lots of TAs. I mean, it's not surprisingly. So this is not the whole group of mine, but it's pretty close. So they will be here in the afternoon, and they'll have badges, and you'll know who they are. So they will help you to get through the tutorials. Okay. So this is for me to switch to to remind you that you have Wi-Fi available here. Without Wi-Fi, it would be kind of difficult to do tutorials. Here's how it works. You on your computer, you connect to Illinois Net Guest, and then yeah, you need to register. You have to do it every day, unfortunately. Correct. And yeah, so you choose. Yeah, you click there. So you don't do this. You click there. And you enter your information. You accept terms and conditions, and then you're online. So they will assign you a username based on your email and a password. That's how you get online. There's also Edge your own here, so if you can just if you have it already available. Any questions about Wi-Fi? If something doesn't work, just raise your hand and we'll figure it out. Yeah. So I think that's it. Are there any general questions about the workshop or where to get coffee, for example? Okay, well then, Shawn, please. Meanwhile, I'll try to see.
Okay, good morning, everybody. Thank you for coming back. We didn't know how many people would sign up. and this exceeded our expectations, or at least mine. I'm here, like Alex said, with Terrell, who you saw in the back of the room, and I just want to start by saying that Terrell really has revolutionized the way that we design DNA nanostructures in our lab. And so a lot of the things that you're going to be seeing is really his hard work. And it's been one of the great scientific collaborations of my career to work with him. And so, I just want to acknowledge that up front. And yeah, he did all the work basically, and then I'm just trying to get the bills paid here. And then the collaboration with Alec is very exciting for us because you guys have such a great center here in Illinois, and we all know DMD and NAMD, and so it's exciting to start to integrate with those tools. So my background is in computer science, not in simulation, and I thought I would start with this sunset from the surface of Mars, because I want to talk a little bit about DNA nanotechnology and the state of the art and where we're going with these tools. So presumably you're all here because you want to make some nanostructure. And NASA has this TRL, or Technology Readiness Level scale, that I thought was kind of interesting. And it has 9 levels. And at the very bottom, when you have a new technology, let's see if my laptop will advance. At the very bottom is basically basic principles are observed and reported. And there we go. Sorry. Here. Something really slowed down on my laptop just now. So let's see if this improves. Okay. Sorry. So basic principles observed and reported at the bottom, and then actual, quote, flight proven through successful operation. So meaning that, the technology that has been on 50 space flights and it works and we trust it. So we can ask, what's the TRL of DNA nanotechnology? And if we trace it back to the beginning, we see that the basic principles were observed and reported, and the technology concept was really formulated at the same time by Ned Seaman back in 1982. And then it took another 9 years before they reached level 3, which is actual experimental function. So they built they show it as a DNA cube. It's really a floppy, a little floppy structure with the connectivity of a cube. So it's not that rigid, but it was a starting point. And it's not every year that a new field is born. So I think that it's pretty exciting that you can still meet the person who did it, and a lot of people that trained with him are now members of the field, of course. I did my PhD with William Shih, and I started grad school right when he was starting his lab at Harvard after publishing this octahedron that he designed, and just actually independent from the field. He just read about this stuff and just worked this stuff out on his own in Jerry Joyce's lab at Scripps. And so I'd say that actually this is still at like level 3 here. Just additional experimental critical functions, figuring out new ways to do things. And origami, so Paul's method to make larger shapes, I think that's still at the kind of level 3. So, and I'll show you what level 4 is in a moment. But there's basically been an explosion of different structures made that are exploring all the different shapes and methods that you can use to build various shapes. So, but they're not yet like building some application. And one contribution that we've tried to make to this is actually move from this level 3 up to level 4. And I would count the postdoctoral work that I did with Guido Bachelet and George Church's lab as something that gets us to the level where we're trying to validate a component of a technology, like in the lab environment. So it's not yet in the real world or in kind of a realistic environment. That would be level 5. This is just like in test tubes and petri dishes. So when we want to really analyze the function of our DNA structures, we do it at that level. And then, but we're still pushing. We still want to go to the next level. And so, and that would be validation in a relevant environment. And then, eventually you get something that really is turnkey and works really well. And, people start companies and stuff like that. So we're trying to push to this level, and that's our focus. And then you can ask, okay, well, what do we need next? And we need real-world applications of DNA origami that solve problems that you can't solve with any other method. Because if there's a simpler method or maybe even a more complex method, but it already works, people will tend to just use those. But then the thing that we've encountered is a lot of the methods were not really good enough to achieve these real-world use cases. So we both have to improve the technology and study it and learn how to build better structures and new structures while we're kind of building the real world use cases. So you kind of have to sail the boat to the destination while you're building the boat is one analogy. So this scale, let me go back to the scale for a moment. So this like straight arrow, that's kind of misleading, right? So for anybody who's tried to develop any technology, you know that you start out with some level or some goal at the top, and then sort of the present state at the bottom. And it's usually not you who started here, but somebody else started there, and they kind of did one level upgrade and then started branching out and trying different things. And so this process, we don't know where it's going to go. Like, this looks really neat and clean at the end, but when we're doing research, we're somewhere in this map. So, and you have to just make a choice. You're like, what do I work on? And then, can I make a bet on this number 4 to try to push it to number 5, or do I need to go back down and like do another number 3 and improve that? So we're always trying to figure those things out. So maybe we think we're going to get to number 5 on this path, but then meanwhile, some other people have been working on their technology, and then they leap ahead. But then you can also solve it in another way, right? So, but then maybe this other path actually gets you other applications. So it's just, the point is, science is messy, and it's not just like climbing a ladder. It's really complicated and it's hard. But then some of us like to work on hard things. And the thing that I think about in terms of what we can contribute is take this messy process, do some part of it, but then also look at like what is actually the thing that worked, and somehow like translate that back into this kind of clean trajectory. And so other people can reproduce that step, and it just becomes reliable. And so that process you can think of as just like writing software, right? We take the things that work and then put them into algorithms and tools and build software tools that people can download and use. And so this is kind of like a little schematic of the way I think about the field and then the work that we do in the lab. And so Terrell has been working on some real-world use cases, and we're not going to talk about them at this workshop, though you can ask him directly about them. He's working on tools to aid in solving challenging structures with cryo-EM, and also patterning ligands to stimulate cells for immunology studies. So these are 2 applications that we think origami is really going to be useful for. And then again, like this slide here, so like he's doing like number 3 and 4 and 5 in this graph, but then also translating the lessons because when you push the limits here, we learn new things about how to improve our tools. So he's basically translating those into software tools that extend what we can do with our design. And so there's a suite of additional tools now that we're calling Cadnano Toolkit. It's unreleased still, so we're working on it, and we'd love your feedback and beta testing. So this is the first time that we have anybody outside of the lab using it, where we haven't helped them directly. So tomorrow is going to go in detail about the algorithms and the different tools that he's used. And he's been building, and we've been working on in the lab. And so you'll learn in detail about that. And then today, for the remainder of my talk, I'm just going to kind of give a history of like where did things come from before this moment, in terms of 3D DNA origami design, and then early Cadnano development, which I'll be the first to admit, Cadnano is not the easiest program now to install or maintain, but we'll try to get something working that matches at least what we do in the lab for you guys. Actually, these are swapped, but I'll talk about just the workshop materials that we've made for Cadnano design. And then I'll just finish, since I have you all here, I'm going to tell you about some new methods from our wet lab in terms of building new custom scaffolds, which you might find useful in the future. Okay, so early work on 3D origami, and where did Cadnano come from? So like I mentioned, I worked with William Shi as a graduate student, and right about the time that I was starting my rotation with him, we learned about this amazing method. I think it was in Albany, New York. He presented it for the first time, and William happened to be at this conference. He said he wasn't really happy to go to it because it was like August. It was super humid and muggy. He was really regretting that he even signed up for it. But then he saw Paul show these amazing data for the first time. And he couldn't even really remember the details of what Paul talked about, but he knew the general concepts. And he was just like giddy with excitement because he had been working for the last 4 years trying to make DNA nanostructures a different way, and it was really hard and slow. And so the really amazing thing, one of the remarkable things, actually 2 remarkable things from our perspective was, one, that you could take the scaffold strand, the single-stranded DNA, and mix it with a bunch of oligos and fold it into a shape, but then you could mix it with a different set of oligos and get a totally different shape. And so this step, this was sort of unprecedented for making such large structures, that you could just, in a matter of a few days, get all the materials that you need to make a new structure, and it could be totally different, not just a small tweak on an existing design. And then this really just scaled quite well. Like you could just keep reprogramming these different structures. So Paul made so many different shapes all in this one paper, and that was just the beginning. That was one remarkable thing. The other that I hinted at is just the scale. These are 100 nanometer size structures. And the thing that maybe you weren't there, it might not be obvious, is that all the structures that people were making at the time were like an order of magnitude smaller. And so this was just such a big upgrade for the field. So we were really excited about it, and we basically just threw everything that we were doing in the trash and started over from scratch when this method came out. So I had been designing structures. William had technicians working for a few years, actually, to put together a library of little pieces of DNA that we were going to stitch together into different shapes using his old method. We didn't immediately stop it, but within a few months, all of that stuff had basically gone into the -80, where I think it's still sitting today. So we saw the paper, or not the paper, but William saw Paul's slides. There was no paper at this point. It was like fall 2005. But we knew we wanted to work on it, so we started developing structures. And the first thing that we wanted to do is just build 3D shapes. So William saw that Paul had made a lot of planar shapes, we know nature builds in 3D, so we wanted to build 3D shapes. And over the next couple of years, working with William as his first grad student, we came up with a scheme to basically pack the DNA helices. We call these virtual helices because they're not exactly a single double helix. They're kind of like a rope, like mini fibers kind of come together to make a single cylinder here. So when I refer to virtual helices, you can just think of that as a stretch of DNA origami helix that has a common helical axis. So Paul had made these planar shapes, and then we worked out that you could actually rearrange the crossovers between the different helices to get quite naturally a honeycomb lattice, and then later we did a square lattice. And so this was really a group effort in the lab. So William and I worked for a few years on this, and then a number of postdocs joined the lab, and everybody was contributing to testing the method and building their own shapes. And so we ended up publishing, like everybody had a different shape they're working on. So we have this grab bag of kind of random weird shapes that don't make any sense. So they made sense in their own context, but we just pulled them all together just to show the generality of the method. And so these are negative stain electron micrographs with 20 nanometer scale bars along with a little model next to it. And so we demonstrated kind of generalized 3D self-assembly with origami, and then we also did hierarchical assembly. So using multiple scaffolds, you can include little sticky end interfaces, and they'll come together to make larger shapes. So here's a wireframe icosahedron, 50 nanometer scale bar there. So again, at the time, we didn't have any software tools. So we had what I like to call author-only tools, like things that the person who wrote it can use it, but then if they give it to somebody else, it is very fragile, it breaks, and it's hard to share. So every origami we made, like, you know, we published several, but each of these were many months, maybe like a full year of effort of, you know, various failed shapes, you know, things that didn't make it into the paper. And so our process, like still today, we had a shape that we wanted to make, and then we knew we would have a scaffold in the lab, and our goal was somehow to generate a list of staple strands or DNA oligo sequences that would fold that scaffold into that shape. And we didn't have really good folding methods or the buffer conditions worked out at the time, or any of the design rules, really. So we were also trying to figure out that stuff at the time. But we had this ad hoc design process where, you know, if we had 3 different lab members working on 3 different shapes, everybody would go, you know, off to their computers and then write some Python scripts and draw some diagrams, and then produce these sets of staple strands. And because our software was so buggy and we were really worried about like ordering backwards DNA, we actually had this buddy system where everybody would have to kind of validate someone else's structure and independently like design the same thing and get the same answer. And so that was that was very tedious. So this is here what our like schematics look like. So precursor to the Cadnano interface. Like I said, we had just custom scripts where we really just look at this diagram and translate that into code. This was obviously very error-prone, very inefficient. And so I wanted to kind of formalize this design process. And so after 2 years of trying to make non-GUI versions of software that do this, it's like I was using input that was like ASCII art. I realized we just needed a graphical interface. I had no idea how to build that at the time, so I just knew I wanted one. So I started working late at night, and just, you know, figuring out like how do you draw something on the screen? How do you handle a mouse click? How do you draw, you know, some text? And so after probably a month of work, the first build of Cadnano was completed on August 3rd, 2007, so over a decade ago. And there's no sense of design. All I had, there's no data structure here really. It's tracking the DNA. I just had the lattice, and then a very simple 3D model. And this was actually running in like Flash at the time. So that was like the Flash browser plugin. So there's a little slider at the bottom that does nothing. And then it's really, I just want to show you that there's like these programs, you know, they have humble beginnings. And so, and one of my favorite stories to tell about grad school is basically how William Shee forbid me from working on this after. I didn't show him this. I knew that wouldn't fly, but I actually worked on this for a few more months. So that's August, and then here's like what I had by November. Let me see if this will play. Here we go. Yeah, so this is starting to look a little bit more like maybe what you recognize. And we were always using pairs of helices, so that was just a feature there. Here's the beginning of just, you know, the strand diagrams. I didn't have a good way to connect kind of the z-axis here to the z-axis in that diagram, so that was just not figured out yet. This is super buggy. It doesn't really work. Yeah, so I think it was even a month later, and then William started to notice that I was getting nothing done in lab because I was just like staying up till 4 a.m. every morning coding this. And so he eventually told me, you know, you should get back to your work, you know, the things that we agreed that you would do. And I said, well, I told him I am working on that. And he said, well, someday somebody will make nice CAD software for DNA origami, but that day is not today. So that really pissed me off. So because I knew like if I just had this interface, I could just draw a structure in like a day. And we were taking, you know, a month at a time, and then we'd hand off our design to somebody else, and then they would design it or kind of try to redesign it. It was so inefficient. It was just like very painful. So I just kind of kept working on it, and then once it got further along, then it came around, and, you know, fortunately. So then we ended up testing it in the lab internally, and then so none of the original shapes were built with Cadnano. But we ported all of them manually, and then the future designs, it helped a lot. And actually, with Hendrik Dietz, for his curved structure paper, we designed like 30 new shapes in the span of a few months. So that really accelerated the design process. So I felt vindicated by that. And William, you know, he was very supportive. He had to come around. So around 2010, I moved over to Wyss Institute and started working with Nick Conway, he's a mechanical engineer, but he loves software. And so he helped rewrite the entire Cadnano data model, and we ported it to Python and PyQt, which is another kind of GUI system. And then at the time, we were making a lot of animations and figures in Maya, and Maya had just updated its interface, so you could actually embed Python and PyQt programs inside Maya and then talk to the Maya API. And so we actually had a nice collaboration with Autodesk at the time, and they gave some developer time to help us build basically a much better 3D view of the origami structures. Unfortunately, we haven't really been able to keep up with the Maya subscription model. So they kind of rewrite the software every year and then break the old version after 3 years. So it's a lot. We don't have a team of dozens of engineers to keep up with this. I mean, or even one really. That's all it would take. But it's quite expensive to do that in a research context. So unfortunately, the kind of Maya integration has broken for a lot of newer releases. And then the way Maya works is it would require a lot of work to continue using it post Maya 17. So in the meantime, our understanding of DNA self-assembly has improved a lot. So there have been a lot of advances. So like Mark Bhatti and actually collaborated with Hendrik Dietz to develop this can-do finite element structure prediction tool. So that was an inspiring work that came out in 2011. She and Yin labs at Harvard have this other kind of staple-only origami they call single-stranded tiles or DNA bricks. Hao Yan's lab has been developing kind of non-lattice or non-honeycomb, non-square lattice designs. And then we've also seen a lot of wireframe mesh methods come out, and then also these amazing multimeric structures. So it's a moving target, and we recognize that Cadnano just needs to evolve beyond the rule-based lattice designs. So I've been working with Nick Conway at the Beast over the last couple of years to kind of refresh Cadnano. And so what you see with Cadnano 2.5 is a new version that has new data structures. It has some new GUIs, but they're not really completed and tested. So what we've ended up doing is kind of relying on the earlier versions of Cadnano and then experimenting with the new GUIs. But I wouldn't say that those are quite ready for prime time. But the key thing is that we have a Python API now. So anybody who wants to dig into this stuff, they can write scripts that will actually interface with the Cadnano design. You can pull all the data out of it, and then even build your own data structures. And that's actually what Terrell was able to build on top of the Python API. The stuff that Alec and Chris did actually predated that, and we've been integrating it. So they were manually kind of extracting information from Cadnano files, which is very tedious. So we're trying to unify all this stuff. So one thing that we did is basically generalize the data model to have 3D coordinates. And so in the old version, it was actually, so here's just a very simple DNA origami design. And it had this fixed data structure, which is really just based on this coordinate system. So this like rows and columns. And then for the honeycomb lattice, it was the spacing, basically the pitch and the size of the DNA strands and where they can cross over is just like hardwired into the software. And in order to have a different lattice, you have to just like hardwire a different set of spacings. And then the crossovers are just sort of built around an idealized form of DNA. The new version actually has 3D coordinates. So if you look at the individual base in the 3D, it would correspond to that position in the orthogonal view, or the slice view that we call it. And then there's an individual base here in the corner. Those all map in the data structure to some position in space. Once we had new 3D coordinates, we needed a new crossover system. And so you don't really need to worry about this. But rather than just having arrays and sort of pointers, we actually now in the data model, it's proximity based. So, and this is, like I said, a new GUI. This is just research right now. It doesn't actually exist in released code that where you can really use this. Well, it's in 2.5. So what I wanted to show is basically you can just drag helices around, and then the possible crossovers actually dynamically update. And so it's sort of like 2.5D. You can imagine there's sort of 3D stuff going on, but then the 2D interface is updating there. And so in principle, this expands the flexibility of what you can make. You can move helices, have them orthogonal, eventually bring the domains close to each other, and see like where to install crossovers. So that's just kind of a future direction and something that we've been working on. We have these different design parameters. So again, rather than just like a fixed array, we can actually say, how many bases per turn is a double helix. So this, you can imagine you're looking down the barrel of a helix, and then each one of these triangles is like a phosphate position, and we can update the pitch of DNA, or like the number of bases per turn, and you can see this updating. So that's gonna, if you imagine kind of twisting or stressing the DNA, over twisting, under twisting it, these locations would move around, and then that can affect how you would design a structure. The data structure, again, was rewritten We have something called a document, which is basically the entire design. And it's this tree data structure. So the document can have a part. And so if you're writing Python code, then these terms will start to be familiar. Like I said, a virtual helix is basically a helix with the common helical axis in the design. So I just put a box around there so that that would be virtual helix 0. We break up we basically house these kind of data structures, a strand set and a strand. So a strand set is basically all the scaffold or all the staple strands. So we keep those, we do the bookkeeping on those separately. And then the individual strands are really a start index and an end index. And then we have convenience functions for, what's the 5' or 3' end of the DNA, or like what is the high or low index. So you can reference all of these. And then we have another parallel data structure going on, which is an oligo. So this is keeping track of the connectivity of strands through the structure. And so this is useful for getting sequence data, for example, or managing the coloring. And so when we start doing Python scripting, what we can do is we import the Cadnano module, read in a file, and then we can do something like ask the document for an active part, and then we can get a virtual helix, and then get the strand set from there. Like the forward strand set is basically going left to right, or in the positive z direction. We can get a specific strand. We can ask that strand for its oligo. We can get the 5' strand of that or the sequence. So you can see how this is going, right? It's like you can really start building your own tools on top of this API. And this is just one example, but you can have coordinates, or there's many things that you can do with this. So now I'm going to just give a brief overview of the tutorials. This is just a couple of slides, so that's part 2 of the talk, very brief. And then the last section will be how we make custom scaffolds, so like new wet lab technology that we're working on. Just want to give an advertisement for that. Because it was the first PhD student to graduate from the lab, and also, again, helped by Terrell. Okay, so tutorials. So I spent a lot of time last week writing tutorials for how to install Cadnano 1 and 2. And we use both of them side by side. And there's some things that Cadnano 1 is good at, like being stable and not crashing. And then there's other things that Cadnano 2 is good at, like quickly editing structures. And so when I make something, I often have both of these interfaces available. And I'll draw things in Cadnano 2 very quickly, but then at some point it often crashes, and you still don't know why that is. At some point, this will have to be rewritten by somebody. But in the meantime, we save frequently there, and then so it's nice to install those. And so we have a tutorial for that for both Mac and Windows, and both versions. And actually, I posted those on my website, or the lab's website, so douglaslab.org/tutorial. So you can already download those PDFs right now, or later today. Another thing that I found useful working on these cloud images is when I'm working locally, I want to be able to swap files between the two systems. And you can set up an FTP server if you're Linux savvy. What I actually found was the nicest thing was just to install the Linux version of Dropbox on the image and run that, and then just set up a shared folder. And so I'll have a tutorial for how to do that for those of you who want to use this. And so this is just showing the image, and then this is a screenshot from my Mac desktop. So I have the same files in both views. And so that's just a convenient way to do things. So the first design, the tutorial, is for a simple, we call it a 10x6 brick. And here's a little movie of the initial structure prediction and coarse grain simulation that the Cadnano toolkit does. And we have an active collaboration, so we're also in the process of like handing this stuff over to the more fine-grained multi-scale simulations and molecular dynamics that Alec does. So you're watching this stuff being built. So that's design one, and there's nothing too special about this. It's meant to be simple, just to get you start to finish through the workflow. The second design is a flat plate structure, and this is similar to a structure that Terrell's been working on, like I mentioned, for one of the use cases. But it's a bit, so the first pass that I made to reproduce this, the structure is not completely optimal, so that will serve as a starting point, and then hopefully we can teach you how we would tweak this structure and really get it very nice at the end. So that's one design. And then the third thing is this wireframe tetrahedron. And so this is, the design process is really involves drawing sketches. So I used Illustrator, so making diagrams, kind of figuring out what 3D shape you want to make, mapping that to 2D. It's a lot of work outside of Cadnano. But then once you have like all the connectivity, you kind of build something in Cadnano and then splice everything together. And so that's the third tutorial. And so hopefully with these 3 designs, that will give you a nice starting point. And then with the rest of the hands-on workshop, we'll be able to go around and tell you how we might help with your, or how we might design different structures depending on what the application is. Okay, so those are the tutorials. And like Alex said, they're very fresh. So, okay, and I'm just going to close with I just have a few more slides left. I just want to advertise some new experimental methods that we've been working on. And so one that we did a couple of years ago was thinking about new design strategies. We were thinking about scaling up origami production. So if we wanted to make grams or kilograms of these structures, we thought it might be useful if we could decrease the number of components. So maybe we don't need to make 150 staples. Maybe we could make 20 staples. So we just asked the question, how could we build an origami by reusing staple sequences? And so we looked at what had been published, and we put them on this little plot here. So this, on the x-axis, we have the number of strands in the design. And then on the y-axis, we have the number of bases in the design, the number of nucleotides. So like Williams' octahedron, it was 1.7 kb, so that's right here. And it was actually 5 strands, so it was like 1 scaffold and 5 staples. So that's not what we called it at the time. And then origami is, closer to 200 strands and about 10,000 nucleotides magnitude. And then you have these wireframe polyhedral meshes, the gridiron designs. So you can see that these are kind of clustering in this area over here. And then the single-stranded tile and brick structures, we have an increase in the number of strands and also kind of a linear increase in the number of nucleotides. And then once you hierarchically assemble structures, so multimerize origamis, then it just goes up linearly. And then the method that we worked on was really out over here. So we wanted like 10 or 20 strands, but the same number of nucleotides. So that means we're reusing the sequences. And I'm just going to give you a very quick overview with this animation. So the standard way that origami works is you have unique staples binding to the entire scaffold. So here we have a little short scaffold and then 12 staples. So when they all bind, you see that this would fold into some target shape. And the concept here was, what if we could have reusable staples, and we actually made a custom scaffold sequence. So we have to actually design the scaffold along with the staples. And so you see, we only have 3 staples here, but then each one of them has to bind 4 times in different locations. So we didn't know if this would work, but we just wanted to try it. And so the idea here is that we're trying to fold the same shape, but using far fewer components. And then So yeah, like that's what we were hoping. We'd see like a similar shape, but we know we only have many fewer strands. And so we were able to build about 6 kilobase long custom scaffolds, and then we folded them from 10, 15, and 20 unique staple sequences. And we actually see it does compromise the quality a bit in terms of if you keep going to fewer and fewer staples based on the design rules that we came up with at the time. But the reason I bring up this project is that we found that we were making a lot of custom scaffolds, and it was time consuming and kind of difficult to make. And so we actually, this is where this kind of zigzag trajectory and working up the ladder here, this was just kind of a thing that we wanted to try, but then it helped us focus what we wanted to do next, which was build a method for making any custom scaffold. And so we just posted this on bioRxiv a few months ago. It's actually now peer-reviewed and out in print, digital print. So you can find this online. And I'm just going to explain how this method works. So one thing we want to do for scalability is use M13 phage. So E. coli together with viruses are these like magical little bioreactors where you can feed them one molecule, and then they'll make hundreds or thousands of copies of that and of themselves. The thing was that it's not so easy to customize the sequence of a virus. So they're usually pretty efficient in terms of the bases that they use, and so if you start messing with them too much, then they stop replicating. So, but M13 is actually one of the pillars of molecular biology in terms of technology. So people have done a lot of really neat and impressive engineering with this system. And so M13 on its own is already really useful for us. So this little animation is basically showing the life cycle of what happens. So we had to kind of understand how M13 works in order to engineer it. And so the way that normally M13 works, it comes in as single-stranded DNA, and then the host machinery it converts it to double-stranded, and then it starts making RNA, a bunch of proteins, and then these proteins come back and process that double-stranded DNA, and eventually start making rolling circle kind of amplification copies of the single-stranded DNA, which then gets exported. So that's what we're starting with. And what we've done for most of our custom scaffolds is basically, just modify the M13 by inserting a piece of DNA. So that can just increase the length by a couple of kb. Another thing that you can do is to get a custom sequence is to basically use a helper phage, which moves a lot of the machinery onto it keeps it on the virus, but then you have what's called a phage mid, which has some of the sequences that are required for packaging of the single-stranded DNA. So here, if you clone that in as a plasmid, so the phage mid's already there, and then you infect it with a helper phage. The helper phage actually starts making, again, all of the phage machinery. And then those proteins, they're hanging out, and they actually start processing this, so they bind to the sequence here on the phage mid. And it includes the M13 origin, so that's basically the signal to make a copy of that, the single-stranded DNA, and then it can be exported if it has the packaging sequence there. So the problem with this is basically this region here is fixed sequence. So the blue region is the custom, but you're still stuck with almost 3,000 bases of non-custom sequence. So our new system, which we call pscaf or pscaffold, has a trick, which basically it has 2 copies of the origin, which includes a start, basically replication start here, and then a terminator here. So the machinery will start here and then stop there, and that will be the entire loop. So you end up with only about 400 bases of a fixed sequence. So if you watch the bottom one, it stops there. So then you get still a helper phage, and then pscaf, and then if you actually use this helper plasmid system, basically there's another system that doesn't get exported, so we combine that with our system. So basically what you end up getting is just whatever sequence you cloned in here, plus just one copy of this, the N13 origin. Okay, so hopefully that made sense. Here's the gel of the different scaffolds we made. So we made 1,500 up to 10 kb length scaffolds. And then we folded those into just simple brick-like structures just to show that it works. And so we deposited this system into AdGene. So those of you who do wet lab experiments, you can contact us if you need any help, and you can already order these plasmids. So those are online. You can read the paper if you're interested. So I think I'm out of slides and hopefully out of time. So I'll just acknowledge our group and our funders. We also do other things like organize Biomod, the student design competition. So if anybody's interested in that, please talk to me next few days. And then, thanks of course to our collaborators, Alec and Chris, who really made this workshop possible, even just by getting together and writing a grant, which we're happy that we got funded. So I think with that, I'll just stop and then take questions.
We have plenty of time for questions.
Yeah. Or if you want coffee.
So that animation you made with the staples pulling together the structure, what did you guys make that animation? That was sharp.
Oh, thanks. With the reusable staples, I think that was Cinema 4D. I didn't make that animation myself. But oh yeah, so I wanted to mention, if anybody needs materials for your talks, like I have animations about DNA origami, folding. You can already visit our lab website, which is bionano.ucsf.edu. And then there's a /downloads, and there's some linked files there, which are just different animations. But if you see me present anything, just email me, and I'll share that with you and share a slide with you, because it's a lot of work to make these. So if you can just reuse them, I think that would be great. You can focus on the unique thing that you guys are doing. Yeah, 3D animation is a steep learning curve. So, but if you enjoy that kind of thing, I kind of like it. So yeah, I could talk to you more about that. But yeah, my slides are a hybrid of Keynote and animations from various different sources.
All right. Is there more questions? If there are no more questions, let's do the following. Let's go to the third floor where you get your badges and coffee. And we'll come back in 30 minutes, okay?